Hello students, welcome to EPG Pathshala. Myself Dr. Ashish Pal, Scientist E at Institute of Nanoscience and Technology, Mohali, Punjab. I am going to discuss a module today that is on nanostructures from protein and peptides under the paper Nanobiotechnology. So the objective of this module is to understand collagen fiber. Collagen is very important for connective tissue, and this gives very strong fibers. So we would look into this hierarchical version of collagen, tropocollagen, to get into ligament or tendon, how the fiber assembles. So these tropocollagens are very unique nanofibers. So we would look into hierarchical version of this collagen superhelix. The next thing we will look at is coiled coil motif. When two helix, two or more number of helix, they come together and they coil together and form this kind of coiled coil motif, and that which is a super helix. After knowing that, we would look into the comparison of both collagen and coiled coil super helix. How is the difference in terms of the fiber formation or many other nanostructure formation? Then we will show how we can also use this coiled coil motif for making self-assembled fiber in a very definite form, or we can also make this self-assembled fiber made from coiled coil motif for as a template to generate nanomaterials like nanoparticle generation in a definite arrangement. At the end, what we will also look at, we will use some other secondary structures like beta sheet forming peptides and beta sheet forming peptides as a head group in an amphiphilic molecule to look at different nanofiber formation and how these nanofibers can be utilized for different applications, something like tissue engineering or drug delivery. Collagen is a primarily structural protein in our body, and it just lies below the epithelium in our body, and it can be in ligament, it can be in tendon, it can be in cartilage, vessels. So actually 30% of the protein in our body are actually made up with collagen. Another important thing about collagen is that the structure of collagen is very much conserved in species to species. That means when evolution has happened, there is not much of change in collagen, and collagen is actually resistant to enzymatic breakdown. That's also very important. And there are actually 28 different types of collagen in our body, and out of that type 1 mostly and type 3 collagen, they are the most stiff collagen. And the stiffness, if you talk about, this can be 100 megapascal. So this is very stiff, and that's what this stiffness, and because of it is made of fiber, it can also have very high mechanical strength, and this is very useful because they are these collagens are in connective tissue, and then they are holding the muscles with the bones. And let's say you talk about our knee, there is a lot of cartilage also collagen. Now if you look at collagen under electron microscopy or X-ray, you do see there are definite band present in collagen. So we would now like to look more in details about collagen fibers. Collagen in ligament and tendon. These collagen fibers are also prevalent in ligament and tendon. So in ligament, what happens is that it connects two bones together, okay, and in the case of tendon, it connects the bone to the muscle, and collagen fiber is integral part of these tissues. And if you look at the hierarchical structure of a collagen fiber into forming tendon, you can see you can have different length regime or dimension. We start with a tropocollagen, which is around 1.5 nanometer, and then that tropocollagen can get assembled in microfibril to subfibril to fibril, and then eventually to tendon. One most important thing in collagen molecular structure is the sequence of triple helix. So normally in this triple helix, there is the sequence glycine XY, where X can be proline and Y can be hydroxyproline. So this triad sequence, they get repeated. And you can see repetitive characters or amino acids of sequence. It can be either glycine, proline, methionine, or glycine, proline, serine. So this is how it gets repeated. Another thing is that now if you look at this glycine XY, this is the amino acid sequence, and this eventually forms this alpha chain of or a polypeptide chain. Now three polypeptide chain that actually form a bundle, and it forms a super helix, or it's a triple helix, and this triple helix is actually known as tropocollagen. And mind it that one alpha chain is actually left-handed here in the case of collagen, but when it forms triple helix, the overall chirality of the super helix is right-handed. And eventually many of these tropocollagen, they actually come together and they form this fibril. One important thing is to note that this triple helix is actually stabilized by the cross-linking between these alpha chains, and which actually this gives rise to very stiff and stable tropocollagen, type 1 collagen fibril. So if you look at this collagen fiber, the electron microscopy, you can see that there are some definite periodicity in the collagen fibers. So this has to do with this arrangement of this tropocollagen in the collagen fiber. You can see that in between two tropocollagen there is a gap, and this is what, when you self-assemble this gap you can see, and this has a 67 nanometer of D period, this is known as. And in fact this particular gap you can utilize for bio-mineralization. So this gap you can actually grow crystals of calcium phosphate, calcium hydroxyphosphate, and this crystals actually strengthens the interactions between the tropocollagen molecules. And this is an example of template driven bio-mineralization. So now we discuss this coiled superhelix. A coiled superhelix is a arrangement where 2 to 7, approximately that number of alpha helix, right-handed alpha helix, they actually come together and form like this rope-like assembly. And this is very important because these coiled interactions, they are useful in gene regulation and many other biological functions. Now important for the coiled structure to form, it's important to have a definite amino acid sequence, which is H, then XX, and then HXXX. So this is important. This is like a heptide. There are 7 amino acids where H is the hydrophobic amino acid and X stands for the hydrocharged amino acid rather, and where the hydrophobic amino acid can be isoleucine, leucine, such type of hydrophobic amino acids. And this amino acids, where they, because of their heptide sequence, they can form a particular arrangement where all the non-covalent amino acids are can come together and can be stabilized. So in the coiled motif, as we are discussing, that we need to have a repeated heptide pattern. In that case it is like HXX, HXXX, where H is known as the position for hydrophobic amino acid and X is for the charged amino acids. Normally these hydrophobic positions are occupied by isoleucine, leucine and valine. So here we see a classical example. The first one, A, is a classical coiled coil of parallel and dimeric leucine zipper, and here we can see the two coils are coming together and forming this coiled motif. There are different representation, and the extreme right one is the representation in red circles. And in the, as you see, this is simplest possible coiled coil, but the lower example is very complex example where it's a complex coiled assembly. A trimer of tetramer is found to be in the structure. When we compare the collagen superhelix and also the coiled coil superhelix, there is a difference. In collagen superhelix, we see that it has a sequence of glycine XY, and XY can be choline and hydroxyproline, or it can be also replaced by some other unit. But in the case of coiled coil, you have heptad, which is HXX. H is for hydrophobic position and X for the charged amino acid position. So HXX, H triple X. So in case of collagen, if you look at the individual helix handedness, the individual helix in collagen is left-handed, whereas in alpha helical coiled coil, individual helix is right-handed. But when you talk about the super helix handedness, the collagen becomes right-handed when you take into account the triple helix. However, the alpha helical coiled coil, the overall super helix handedness is left-handed. We will see how coiled coil motif can be useful for assembling nanofibers. So we take, for this reason, we choose two polypeptide chains which are complementary to each other. That means they have complementary glutamic acid and lysine unit in certain positions, and this has been chosen to have this heptad sequence where it is HXXX, HXXX. That's the sequence it is running, and it has been designed such a way that it has got a sticky end in terms of the electrostatic interactions. And through the Weng-Chiang diagram, you can see this A and D has been only populated with hydrophobic amino acids in both 1 and 2 polypeptide chain. That this lysine and glutamic acid, they are, you see this arrow, that means the arrow shows that they are electrostatically interacting. That's how they can form fiber. However, there was a control experiment where there is no sticky end. For the case, if you take another polypeptide chain, that is 3, there is no sticky end possible, and 2 and 3, when they form assembly, they can form no sticky end. It's like a blunt end, and this has been shown. They form these self-assembled fibers. See, this coiled coil motif is a computer generated model. This coiled coil motif form very nice nanofibers. Similarly, this coiled coil motif can be also used for assembling gold nanoparticle. So in this case, they again, there are two polypeptide chains A and B. They have been taken, and they can form again very nice belt and brace like peptide system. And this is where you can actually, in that junction, you can actually synthesize gold nanoparticle, and you can see the TM image of three-dimensional gold nanoparticle that is assisted by this self-assembled peptide fiber. The beta sheet and polymer hybrid for assembling nanostructure. So if you look at this design of this molecule, there is one side you have polyethylene oxide chain, which is hydrophilic in nature. On the other hand, you have valine-threonine like this peptide sequence, which are known to form beta sheet assembly. Because of, we see that if you look at this TM image, they form very nice peptide nanofibers. And when you look into the cartoon of proposed model for this assembly, we can see this peptide unit, this beta sheet, they can form antiparallel beta sheet, so that the core, so basically each fiber can have two stacks of this polymer beta sheet hybrid coming together, and it can have the core which is hydrophobic peptide in nature, and the polyethylene oxide will give it solubility. Polyisoprene based hydrophobic block was functionalized onto oligopeptide block, and based on the combination, 8 different molecules were actually made as shown in the slide. And you can see compound number 1, 2 and 3, where you have one diacetylene unit, and one side you have polyisoprene unit, then the beta sheet forming peptide unit. The other side of the diacetylene, you have one amide bond forming unit present there. But if you look at compound 4, this has diacetylene group flanked by both the side polyisoprene group and beta sheet forming oligopeptide group. In the compound number 5 to 8, we have only one side of the diacetylene has oligopeptide and isoprene block, but the other side there are polar groups present in there. The AFM image shows that compound 1, 2 and 3 very long fiber with high aspect ratio, and these fibers have parallel beta sheet domain with very pronounced twisting in the fiber. But for the compound 4, there was no twist observed and only flat tapes were observed, which comes from single parallel beta sheet unit. These different molecules, they can be categorized in 3 different classes. One in case 4, where both the side of diacetylene is flanked by this polyisoprene block and also the oligopeptide block. This eventually has both the side 5 and 5'10" hydrogen bond form, and this form tape-like structure through parallel beta sheet domain with opposite orientation. In the case of 1, 2, 3, they have 5 plus additional another hydrogen bonding from the other side, and they form a single parallel beta sheet like structure. They eventually can form a ribbon because this each one they have each tape, they are helical in nature and they can actually twist around each other and form this helix bundle, double ribbon like structure. But in the third case, you can have only single beta sheet which is anti-parallel tape like structure, and they form flexible thin tape with featureless cross section. So now we look at one peptide block copolymer, Amphiphiles. So this is an example from the group of Samuel Stoop, where he has functionalized one fatty acids with different blocks of peptide. Here you can see that there is a block where you have a lot of cysteine unit present, then there is a block where you have flexible glycine unit present, and then followed by a fourth block where you have phosphate group, and then you have a charged group present over there. And this is called a peptide amphiphile because you have the head group amphiphile is peptide, and this forms very nice fibers. You can see from the cryo-TEM, these are all nanometer in dimension, so you can have very structured formation of the self-assembled nanofibers. How you can change the molecular structure in the peptide block, and you can actually get the macroscopic property which is completely different. So it is more structure property relation study, where the Samuel Stoop group, they have made peptide amphiphile, and another case is very soft peptide amphiphile. But in the case of stiff peptide amphiphile, you can see all the peptides, they are connected in their backbone. But in the case of soft peptide, you can see one of the lysine side chain, and in there that this amino group has been used for peptide bond formation, so that there is a kink. So they introduced the kink in the head group, and because of the kink, they suggested that this will not pack in a very nice way, and there will be always curvature when you make the peptide. And indeed, they see that in the case of stiff PA, that is the first one, polypeptide amphiphile are expected to have very strong beta sheet and they form very stiff nanofiber. But in the case of soft peptide amphiphile, they are supposed to have weak beta sheet interaction, and because of this branching, they form flexible nanofiber. They can also see from the CD experiment, they could see in the case of stiff peptide amphiphile, they saw very nice beta fiber, but in the case of soft peptide amphiphile, this was not a very distinctive signature of beta sheet, rather it was like a little bit random coil based structure. However, they applied these nanofibers in tissue engineering, and what their objective was that they wanted to grow neuronal and differentiate into neuronal cells. And as you know, in the case of neuronal cell, the cells will be little bit high aspect ratio, and they will have fibril like morphology, and they see when they incubate the stem cells in different peptide amphiphiles. In the case of soft peptide amphifile, they get differentiated into the neuronal cell. But in the other case where they have used stiff peptide amphifile, that case after 28 hours they develop astrocytes. peptide amphiphils with peptide fragments IK, VAV fragment that can also be used for neuronal cell differentiation. In fact, this peptide, they form very nice hydrogel DMEM cell medium. You can see the nice fibrous structure from the ACM images, and this C and D, they show the cell culture media and cerebral spinal fluid, where this particular IKVAV, they form hydrogel solution. So this slide shows that IKVAV peptide amphiphile gel within 7 days, that gives very nice neuronal cell differentiation, which is characterized by beta tubulin expression as compared to laminin or other system. But you can see that for laminin and other system, it is mostly the astrocyte or glial cell that gets expressed. And there is also important that how much percentage of IKVAV containing molecule should be doped into the gel. Then you can see that beyond 40% of that IKVAV based polypeptide amphiphile, you can have better cell differentiation of the hydrogel medium. So let's summarize what we have learned from this module. So first of all, we have got to know about collagen and how the collagen from triple helix, rather the hierarchical structure of collagen fibers. Like if you form tropocollagen, then you eventually move up there, and triple helix of collagen you can form. The gap between the tropocollagen fiber, how that can be used for bio-mineralization. So we understood collagen. So after that, after knowing collagen, then we move to coiled coil motif, and the coiled coil, we also compare both coiled coil motif and collagen, how these two structural motifs are different, because many people tend to confuse these two superstructure of let's say collagen or coiled motif. So we categorically looked at different aspects of these coil and collagen. Then eventually we looked more detail also about coiled coil motif and what are the sequence you need to have coiled coil motif, and the heptide sequence particularly, and how heptide sequence can be actually represented from 3 dimension to 2 dimension projection, so that in order to understand that better. And then then there is again the sticky end concept for this hydrogen bonding, which can come out from this coiled coil motif, that we also found out, and with that we also designed peptide nanostructures, and eventually using this coiled coil motif or peptide nanostructure, it's like a belt and brace, we also could assemble nanoparticle in a particular dimension. Then lastly, we also looked at how to design nanostructures coming from the beta sheet assembly or helical assembly, and eventually they form fibers, and that can also form hydrogel from cross-linking. We have looked into different examples, and then looked at how changing the conformation in the peptide sequence, how you can tune the fiber morphology and eventually the mechanical property of the fiber, which can actually reflect in the cell differentiation using that hydrogel scaffold. So I hope this entire nanostructure designing using the peptide sequence would be helpful for the students. Thank you.